Good afternoon, Francisco. We are going to talk about a biological topic that is the reproduction of a cell. I'm going to talk about meiosis. Meiosis is the process in which an eukaryotic cell separates the chromosomes in its cell nucleus into two identical sets, into two separate nuclei. Phases of the cell cycle and meiosis. The first one is the interphase. The meiotic phase is a relatively short period of the cell cycle. Interphase is divided into three phases. The first one is the first gap, or the G1. The second is the synthesis, or S. And the last one is the G2, second gap. During all three phases, the cell grows by producing proteins and cytoplasmatic organelles. However, chromosomes are replicated only during the S phase. Now I'm going to explain the prophase. The nuclear membrane disintegrates and microtubules have embedded into the nuclear space. These microtubules can attach to the kinetochores or they can interact with opposing microtubules. Metaphase. The chromosomes align at the metaphase plate. Now I'm going to explain the anaphase. The chromosomes split and the kinetochore microtubulus gets shortened. The last one is the telophase. The decondensing chromosomes are surrounded by nuclear membranes. Cytokinesis has already begun. The pinch area is known as the cleavage furrow.
Now I'm going to explain the process of meiosis. This is very similar to mitosis, but more complex. While mitosis produces two daughter cells from each parent cell, meiosis results in four sex cells, or gametes. Unlike the cells created by mitosis, gametes are not identical to the parent cells. They are genetically different. They are used then in sexual reproduction. Meiosis allows the daughter cells to have one half of parent cells. It means that it has one half of chromosome number, because when the gametes unite to form a zygote, they will have the correct number of chromosomes. Now here we have the cell nucleus, which has chromosomes from parents. During meiosis, chromosomes from both parents are copied and paired to change portions of DNA. First, the chromosomes replicate, and then the homologous chromosomes pair up. Then chromosomes swap sections of DNA, and this creates a mix of new genetic materials in offspring cells. The process of meiosis is divided into two, and first we have the meiosis 1. That is when the chromosomes pair divide, and is that the nucleus divides into daughter cells. And then is the meiosis 2, when each daughter cells divides again. So chromosomes divide and daughter nucleus have single chromosomes and a new mix of genetic material.
Hemophilia is a recessive genetic disease associated with X chromosomes that consists in the difficulty of blood to clot properly. For deficiency in a protein called antihemophilic globulin coagulated. The two types are hemophilia A, there is a deficit of coagulation in factor 8, and hemophilia B, there is a deficit of coagulation in factor 9. In each cell, there are 46 chromosomes, half resided from the mother and half from the father. Chromosomes come in pairs, so you have two copies of all your genes. But there is one exception: the sex gene X and Y chromosome. The X chromosome have genes for producing the factor 8 and factor 9 blood coagulation related. If a male inherits a chromosome with a damaged gene factor 8, there is not a copy of this gene so it cannot produce the clotting factor. These injuries normally occurs in women, but in very low percentage. People with hemophilia can bleed after injury or accident for a long time, and they may also bleed from knees, elbows, and ankles. Brain hemorrhages are a very serious complication of hemophilia, and that needs emergency treatment. The main treatment is the injection of the missing factor clotting in the bloodstream. Down syndrome. The Down syndrome is a genetic disorder caused by the presence of an extra copy or a portion of chromosome 21 instead of the usual two, characterized by the presence of a variable degree of mental retardation and peculiar physical characteristics that make recognizable persons. The syndrome is produced by the appearance of one or more in original chromosome 21 in the cells of the organism. In this genetic material, which cause more physical features and developmental delays associated with Down syndrome. The 95% of cases show the excess chromosome to an error during the first meiotic division, and call it this variant trisomy 3 or regular. The error in this case is due to an incomplete disjunction of genetic material from one parent. Not completely understood the causes of erroneous disjunction, as in similar processes are multifactorial hypotheses proposed, as environment, exposure, or cellular agent. However, there is no established relation between any causative agent to the syndrome. The only factor that has a stable statistical relation or association with the syndrome is the maternal age. So that is why there are complications in babies born to mothers over 35 years, and no matter the age of the parents.